Oh, I'm so glad to see such a wonderful and lively audience, and we're really delighted to have you here this evening. I'm Rick Lifton, the president of Rockefeller University, and I'm really thrilled to welcome all of you to the presentation of the 2025 Lewis Thomas Prize for Writing About Science. Tremendous scientific discoveries are made every day across the world. However, due to the increasingly complex nature of research, there are inherent difficulties in translating scientific concepts to a lay audience through dense, peer-reviewed articles and papers in the scientific literature. This can, at best, perhaps alienate the general public, leading to misunderstandings and mischaracterizations of our discoveries and the benefits to humanity. Now more than ever, it's imperative that scientists bridge this divide, finding ways to convey the excitement and beauty of their work to everyone, not just to one another's colleagues. Rockefeller's Lewis Thomas Prize was established in 1993 for this purpose, to recognize and honor scientists who, through their writing, effectively bring scientific knowledge into public discourse, particularly those whose writing also evokes reflection, revelation, and wonder at the natural world. The prize is named for its first recipient, Lewis Thomas. His book, The Lives of a Cell, Notes of a Biology Watcher, a collection of insightful essays on science, nature, and human biology, received the National Book Award for nonfiction in both the arts and letters and science categories in 1975. The Lewis Thomas Prize has been awarded to scientists whose writing has illuminated a wide range of fields. These include molecular biologist François Jacob, cosmologist Martin Rees, sociobiologist E.O. Wilson, psychologist K. Redfield Jamison, mathematicians Ian Stewart and Stephen Strogatz, oncologist Sid Mukherjee, oceanographer Sylvia Earle, psychologist Jennifer Eberhardt, and I could go on. The Science Prize, these recipients are selected by a jury including distinguished scientists and authors. This year's prize jury was chaired by Jesse Ausubel, with members Diane Ackerman, Jan Breslow, Devin Collins, Anna Fells, Tom Sackmar, and Jonathan Weiner. And I'd like to thank Jesse and the selection committee for their efforts in selection of this year's prize winner. Thank you so much for your efforts. And this year they came up with a real show-stopping star. And it's now my pleasure to introduce this year's recipient of the 2025 Lewis Thomas Prize, Dr. Stanislas Dehaene. So, Stanislas Dehaene was born with a gift for mathematics. When he was 5, he intuited the infinite sequence of whole numbers. And after counting all the way to 1,000, he felt the thrill of realizing how large numbers could actually be. His father was a pediatrician and worried that local schools in the small industrial city of Roubaix in France, where the family lived, lacked the enrichment that his son needed. So he fed young Stanislas his insatiable curiosity with books and magazines like Scientific American, National Geographic. And one evening in 1979, he came home with a big box, and inside was one of the first personal computers, a Tandy TRS-80. I'm sure all of you remember that machine. So once it was set up in the family's dining room, Dehaene, now 14 years old, could often be found in front of the keyboard typing out lines of text, numerals, and symbols as he programmed in the early code called BASIC, invented in fact by John Kemeny at Dartmouth College, where I was an undergrad. And so I remember those days of BASIC, not as well as you do, I suspect. But one day, he simulated planet trajectories as they go around the sun. And 4 years later, in 1983, he matriculated into one of France's elite universities, l'École Normale Supérieure in Paris. After earning his undergraduate and master's degrees in mathematics and computer science, he realized that although he loved math, he was particularly interested in figuring out how the brain does math. So he decided to get a PhD in cognitive science and found himself working in the lab of Jacques Baillière, a leader in this burgeoning field. And it was during this time that he began a lifelong quest to understand which mathematical concepts are innate and which are acquired through learning. So he began attending the lectures of France's most famed neuroscientist, in fact, a 2004 Lewis Thomas Prize recipient, Jean-Pierre Changeux, who wrote the bestseller, The Neuronal Man, The Biology of the Mind. Now, parenthetically, many in this audience will know Changeux as well for his contributions to biochemistry via the fundamental concept of allostery, that altering the critical activity of a protein by a chemical's binding to a site remote from, say, the catalytic site of an enzyme, or another chemical such as carbon dioxide, how its binding induces hemoglobin to release oxygen to tissues. But I apologize for digression, so back to Changeux, the neuroscientist. So after one of Changeux's lectures, someone handed Dehaene a small piece of paper, and on it, Changeux had scribbled, "Please meet me after class." Well, in fact, he was looking for a mathematician to create a computer model to simulate how humans estimate numbers. Dehaene found himself between two warring factions. Mailer was looking at how the brain's cognitive functions work together without caring exactly how those functions were implemented inside the brain. Changeux wanted to map the specific neuronal networks that gave rise to thinking. Dehaene decided that his career goal would be to reconcile those two. This spark of a hopeful approach to the problem came in 1988 when he learned about an early version of positron emission tomography, or PET scanning, being undertaken by American cognitive psychologist Michael Posner and colleagues in Oregon. Realizing that this technique could transform his work, he completed his PhD, then headed to Oregon as a postdoc to study brain imaging with Posner. Dehaene put his new skills to work when he returned to Paris in 1995 and took a job with France's National Institute of Health and Medical Research. In those days, brain scanning took heroic efforts. Think of chemists racing down the hall with radioactive tracers in syringes, delivering them to Dehaene so they could be administered before the tracer decayed too much. It was a far cry from the extremely powerful 11.7 Tesla MRI instrument that he now uses as director of Neurospin, France's Advanced Brain Imaging Research Center, and as chair of experimental cognitive psychology at the prestigious Collège de France. Dehaene's experiments, performed with numerous co-authors, have produced fascinating revelations. He has found regions of the brain that contain pre-programmed circuitry for estimating numbers and for responding to language at birth, and showed how formal education can expand and change the brain's capacities. He also learned that consciousness arises only when a certain threshold of neural activity is triggered. With each milestone of accumulated knowledge about the brain, in addition to his brilliant next chapter of science, he began writing books about these discoveries for a wider audience on topics including the brain's innate number sense, how humans learn to read, and the neural basis of consciousness. He brings together many insights from those earlier publications in his book, How We Learn, Why Brains Learn Better Than Any Machine, for now. In it, his lively writing, humor, and adept metaphors convey a feeling of awe about the miracle of the brain's plasticity and how it leads to learning, and all of which done in a way that still allows plenty of room for discussing at depth the scientific underpinnings that led to these new insights. It's a remarkable combination. And this approach gave way to more philosophical musings on humankind's quest to understand the brain in his most recent book, Seeing the Mind, Spectacular Images from Neuroscience and What They Reveal About Our Neuronal Selves. For me, this book is remarkable for its fascinating content and for its unique organization, which I really think is innovative. The book comprises nearly 100 couplets of 2 pages. The left-sided page, a stunning image illustrating a fascinating aspect of the brain's organization or molecular function, with the facing page on the right providing a very succinct description of the major concept in neuroscience that's illustrated on the facing page. This innovative presentation is perfectly tuned to our increasingly short attention spans and readily distracted lives. He successfully conveys a coherent sketch of compelling concepts in brain biology in just a few minutes, one bite at a time. It's both beautiful and inspiring. Tonight in his talk, Seeing the Mind, Past, Present, and Future of Brain Imaging Research, Stanislas will showcase the exciting progress of brain imaging and recording techniques that allow us to visualize the human brain from entire cortexes down to single cells. We will then open the floor to questions from the audience, and I'd now like to invite Stanislas to please join me on the stage. Well, they're way ahead of themselves. You'll get another one of these. So it's now my great pleasure to read the citation for this year's Lewis Thomas Prize. Throughout his career as scientist and author, Stanislas Dehaene has drawn upon his deep curiosity, keen intellect, and prodigious imagination to elucidate the inner workings of the mind. He has pried open and explored the black box of how we learn and conveyed his insights through clear and engaging prose. His writings have introduced readers to the subtle logic of the brain's intricate architecture with its specialized neuronal locations and structures that are primed from birth to respond to faces, language, numbers, locations, subliminal messages, and more. In his fourth book for the general public, How We Learn, he chronicles his experiments to show how the brain from a tender age uses reasoning, abstract concepts, and systematic rules to model the world. He details the interplay of nature and nurture in constructing the adult human brain and offers science-based principles to help us appreciate how a child's cognitive development is transformed through learning. Seeing the Mind, his most recent book, takes the reader into a visual realm with stunning images from delicate multicolored branches of cortical neurons to hauntingly beautiful layered brain silhouettes that illustrate critical differences between early humans and Homo sapiens. The images are paired with one-page essays that are packed with science and peppered with philosophical musings that narrate how our comprehension of the brain has evolved over time. Dr. Dehaene has undertaken bold creative research that ultimately seeks to understand human singularity. He generously shared his journey with us in hopes we will grasp and embrace that we are, in his words, a small miracle of neurobiological computation. His drive to demystify the mind while preserving our sense of wonder and his rare ability to educate and enthrall through the written word exemplify the qualities we seek to recognize with the Lewis Thomas Prize. Dr. Dehaene, it's my great honor to present you the Rockefeller University's Lewis Thomas Prize for Writing About Science. We congratulate you on this award.
Dear Professor Lifton, dear members of the committee, dear colleagues, ladies and gentlemen, I can't even begin to tell you how honored I feel to be awarded the Lewis Thomas Prize for Writing About Science this year. As you can hear, English is my second language by far, and receiving such a distinguished prize for my writing in prestigious Rockefeller University is truly a privilege. When I look back in time, it seems that my entire life was nurtured by good science writing. In my teens already, as was mentioned by Professor Lifton, my father would feed my appetite for science with monthly issues of Pour la Science, which is the French edition of Scientific American, where I devoured articles, in particular by Martin Gardner's. At the age of 16, I was exposed to Jacques Monod's Le hasard et la nécessité, Chance and Necessity, and also to a book that became my scientific program, Jean-Pierre Changeux's L'homme neuronal, Neuronal Man. And these books, followed by the reading of such giants as Richard Dawkins, Dan Dennett, Jared Diamond, Steve Pinker, Oliver Sacks, many of whom are previous recipients of the Louis Thomas Prize, shaped my life as a scientist and as a writer. And I'm delighted to now join their distinguished group. And later at dinner, with the help of a little wine perhaps, I might tell you how great science writing also shaped my family, because I met my wife in the Algerian desert on camelback because she was reading Stephen Jay Gould's Panda's Thumb. But that will be off the record. So tonight I want to tell you about what is probably the most complex object in the universe, a machine whose efficiency is unmatched by any artificial intelligence so far, the human brain. How the brain works, how it generates ideas, decisions, thoughts, and feelings is the quest of my life. It's not finished. And I want especially to ask how its abilities grow with education, and perhaps why do they shrink with politics. I won't talk much about politics tonight, but I want to cite the humorist Ambrose Bierce and his famous Devil's Dictionary, which provides a very up-to-date definition of the brain. I quote him, an apparatus with which we think that we think. And he continues, in our civilization and under our republican form of government, I'm just quoting from Bierce, under our republican form of government, the brain is so highly honored that it is rewarded by being exempted from the cares of office. Okay, so understanding the human brain is one of the greatest challenges of the 21st century. We are pretty sure that its circuits are responsible for every thought and every feeling that we entertain, but we don't quite know how. And the challenge is actually nicely illustrated by this engraving, which is from the 16th century by Matthias Grother. And on it, you can see a person lying in which is actually an alchemist's oven. And out of the oven comes all of these ideas that the person has in mind, including perhaps his mistress or the color of his underpants or something. But with our modern eyes, it's hard not to see someone being scanned in MRI, perhaps. And that's exactly the challenge. You know, can you read from the brain what is the person thinking? Could you understand the brain well enough to understand this rather remarkable mapping between the mind and the brain? So I would like to describe to you today some of the progress that is being made in this area. And at the end, we will ask about the most recent challenges. There are several steps on the way, and I won't show them all, but this is one very important step. The first time, perhaps, where there was a non-arbitrary theory of mappings between cognition and brain. This is the original brain of a patient, Tantan, Le Borgne was his name, the patient from Paul Broca. You can see that he has a lesion, is what is now called Broca's area. Broca noticed that the patient was suffering from inability to speak, to have articulate speech. That's why he was called Tantan, because the only thing he would say was tan, 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 tan. He could not say anything else except for a curse word, sacré nom de Dieu, okay, which probably means there is a different circuit for that. But so this was the first time where a concrete proposal was made on a solid empirical basis for the mapping between specific brain areas and particular functions such as articulated speech. And this is actually a rescanning of that brain by Nina Dronkers and collaborator, vindicating the finding, but also finding that the lesion is actually going deeper than we thought. But the next step, as was kindly mentioned by Professor Lifton, for me is this very important date of 1988. You know, people talk about flashbulb memories. I'm sure you have some of the 2001 attack. Well, I have a flashbulb memory of reading this paper. I know exactly where I was and what I was doing. This is the 1988 paper by Michael Posner and collaborators, especially Marcus Reichel. They were using PET scanning to show some of the mappings between the brain and cognitive functions in the domain of language. They could see that when you were hearing words or seeing words or speaking, but especially when generating words, there was intense activity. It was tightly localized to specific sectors of the brain, and this is, of course, in the vicinity of so-called Broca's area, the ability to generate words associated with stimuli. This was, for me, a revelation. The program of Neuronal Man was just not an abstract program. It was doable. It was feasible thanks to hierarchical designs like this and clever machines to actually map which brain areas were doing specific functions. And when I saw this, I knew what I had to do in science. That was already a revelation. Now, you have to realize what this involved. It did involve having a cyclotron in the basement, generating radioactivity that would decay in less than 2 minutes by the half-life. So you had to be really fast. This is an image of an average brain. So you have something like 20 subjects going into this picture, and you could only get one image every 12 or 15 minutes because of the radioactivity having to decay before the next injection. So fast forward to modern brain imaging, it doesn't require any injection, no radioactivity, just lie in the scanner. And this is, of course, MRI, functional magnetic resonance imaging. This particular image comes from the lab of Jack Gallant and Alexander Huth in Berkeley. What you can see here is now a single person, not an average brain, very high resolution so that you can map the function of individual voxels on the surface of the brain. And in this case, it's a mapping of meaning onto the brain. The details are still a little bit controversial, but you can see on the right that for every voxel that is present on the brain, or for every element of surface of the cortex here. By the way, the surface has been inflated. This is the original folding, but you inflate it in the computer so you can see better inside the folds. But the colors indicate different aspects of meaning. And basically, we all have maps of meanings onto the brain, such that, for instance, this particular region seems to respond to social knowledge. And these are the words that will trigger the activation of this region. So you no longer need to average across subjects. You can have single subject resolution. With MRI, you can get images every second or 2 seconds, and you get, therefore, a whole dynamics of brain activity. This is becoming, the workhorse of people who want to understand how the brain works. What is the future? The future is perhaps bigger magnets. There are many directions this research is taking, but this is the arrival in Eurospin, the building that I direct, of the Isolt magnet, which is an 11.7 Tesla magnet. This is the largest magnetic field that has been achieved so far for the human brain and body. The difficulty is, of course, to have a magnet that will fit a body inside, which is 90 centimeter diameters. And this machine weighs 150 tons. It is a giant in all sorts of respects. It has special supraconductors. It has to work with lower temperature than usual, so it's cooled with liquid helium, which is at 1.8 Kelvin, so that it is super fluid. All superlatives could apply to this machine. But you see, this is 2017 when it arrived. And on my next image, it's 2023, the very first images. We really depend on the whole army of engineers and physicists in order to make such a machine work these days. And you get an idea of the sort of images that we're hoping to get with new machines like that. This is like getting a new telescope. So on the left, I hope you can see that the image is not bad at a classical 7 Tesla, already quite top of the shelf machine at 7 Tesla, but it's a little bit noisy, and you get exquisite details of anatomy from a higher magnetic field. Basically, you get better signal in all respects. So if I zoom in on the occipital region here, you can see that it's possible to distinguish some of the layers of the cortex in fine detail and to see some of the boundary between areas. And of course, this is just the anatomy of the brain that we are getting for the moment. We are eagerly waiting for the first functional magnetic resonance imagings of the brain. You have to realize that MRI is extremely flexible and versatile, so there's a lot you can do with it. You can get the anatomy of the brain. You can get the connectivity of the brain. This image is an image where the colors represent the direction of the fibers of the brain. This is a magical technical diffusion MRI, where what you measure is the diffusion of water molecules and the direction in which they are moving. And it turns out that if you are in the white matter of the brain, which is full of fibers, the water will move along the fibers much more easily than it moves perpendicular to the fiber because of all the membranes that are blocking its way. And so what you see here is the direction of the motion, and from that we can reconstitute the highways of the brain, the connectivity of the brain, how information is carried from one place to the other. This is still anatomical information, but of course I want to go back now to function of the brain, and this is now back to 7 Tesla images because we don't yet have 11.7 Tesla images, but these are images of things that we can very easily do in the lab these days. These are 3 different people who went for a short scanning session of 1 hour, and in fact, in just 5 minutes, you can map these different areas of the brain that respond to specific categories of picture. So all of you in this room, you have areas that care about faces, especially in the right hemisphere here. You have areas that respond to bodies next to it, to places more mesially in the brain, and I studied a lot this region that responds to written words, which is the result of education. Because you've learned to read, you have this area of the brain that responds to written words. Now, this is still an indirect measurement, right? We are measuring oxygenation of the blood that is irrigating these areas of the brain. So we're not directly seeing the neuronal activity with these techniques. So how do we know this is real? How do we know that there are actually sectors of the brain that care only about places or only about faces? Well, we are lucky enough to have occasional cases of single neuron recordings from similar areas. This is from the work of Florian Morman a few years ago. And you can see here, for different pictures, the response of a single neuron in the human brain in an epilepsy patient. Of course, this is not done in the normal human brain, but only for clinical purposes or as a side exploration of clinical patients. And you can see this neuron reacting anytime there is a place on the photograph, regardless of the particular place in this case. So this is a neuron that comes from this parahippocampal place area. And you can see it will not respond to people or to animals or to objects unless there is also a background where there's a very clear place, which is mentioned in the figure. So this is a place coding neuron. I strongly believe that progress in our field will come from this ability to record more from single neurons and actually from collections of single neurons so that we can understand at the neuronal level the neural basis of cognition. And to some extent, progress is being made. This is from the work of Rodrigo Cuyan Quiroga, who already a while ago was discovering concept cells. I'll let you figure out what this neuron is responding to. You can see the discharges here, and I hope you can see, I think you all know this character, right? I mean, he's not real, but we have a representation of him in our minds. And this particular neuron was responding to Luke Skywalker, regardless of how Luke was presented as a picture, whether he was young or old in a movie, as a name written on the screen, or as a name spoken by a male or by a female person. We all have in our heads cells that are ultra-specific to specific people, specific places, specific concepts, and don't react to equally famous people, other names, places, and so on. They will react only to one particular concept. Almost one. I don't know if you've noticed this particular guy here. This is Yoda, right? So why is this neuron also responding to Yoda? Nobody knows. These are quite heroic experiments where the recording is quite short. So maybe the neuron is responding to all Jedi. That's one possibility. Or maybe Yoda is evoking Luke Skywalker and the guy was actually thinking of Luke Skywalker. We don't know. So this is the limits of these experiments. But I think there is a really nice work to be done to understand what these units are doing in the human brain. And I just wanted to tell you a little bit of recent science, just a paper that was published a few months ago, where we used the existence of these concept cells to ask whether there is actual language processing going on that we could see in the same concept cells. So here's the location of several neurons in the hippocampus or near the hippocampus. In this particular case, it's a neuron that responds to Shrek. Don't ask me why. You know, these are movie characters, but this neuron responds to Shrek and not to, let's say, a family member of the patient. Okay, and you can find the converse, of course. The question we asked is whether these neurons would respond only to the presentation of the actual person, or would they also respond to a pointer to the person? And the pointer in language is a pronoun, or can be a pronoun. So suppose you hear or you read, Shrek and Courtney Love went into a bar. It sounds like a joke, right? But he sat at the table. He can only be Shrek because Courtney Love is female, right? If it was she, it couldn't be Shrek. And if Shrek was not mentioned, but maybe Barack Obama went into the bar, then he would be Barack Obama and not Shrek, right? You get the idea. So he can be a pointer that specifically refers on a very local basis to a particular person. So the question we ask is whether there would be reactivation of the neuron just by the pronoun he. And this is exactly what happened. You see the initial response to Shrek walked into a bar, and then he sat at the table, the neuron reactivates. If it is she, it doesn't work. And if Shrek was not mentioned in the original sentence, it doesn't work either. Okay. So this neuron is a pointer to the corresponding concept. This is what you get out of averaging 53 hippocampal neurons, so the signal is clearer relative to the noise. And even better, I think, is you can apply decoders. So you can train a machine learning algorithm to distinguish between these three possibilities for the antecedent of the noun. And you see that you can decode whether the noun is first or whether the noun is second in the phrase, but you can very strongly decode what is the name which is referred to by the pronoun. The level here is 1, which means close to perfect. Now, from a small sample of 53 neurons, you can decode quite nicely what was the referent of the pronoun. And you can also decode errors that the subject is making. So I'm just using this as an example of the forefront of research. This is an example of a neuronal pointer, or the result of pointing in the human brain. And it is absolutely necessary for the kind of machinery of linguistics that linguists have been postulating for many, many years now. We don't know how it works at the neuronal level. I believe that there are many neural codes, perhaps some that are completely unique to the human brain, unique to human symbols, that remain to be discovered. And we have to get better in looking at single neurons. Now, I want to speak a little bit about the impact of education on the brain. There would be no neurons responsive to Luke Skywalker if you could not read the word Luke Skywalker, right? Or if you had not seen the movie. So this is the result of education. But we decided with my wife, who is the author, the first author of this work, that we should study this right from birth. And it's been a very interesting quest to see that learning starts very early on, that language, spoken language, starts very early on in the baby's brain. Written language is, of course, an invention, but spoken language is something that the brain is wired up to learn very, very quickly in the first few months of life. So we started to do this with this interesting contraption. Here it's called an geodesic electrode net with already, I think in this case, 128 electrodes, initially 64. And you can see some of the basic paradigms that were used. If you present to a baby ba, 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 the brain will habituate. The response will go down and down and down. But if you present to the same baby ba, 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 ga, and the response of the brain will increase, not the initial response, but a little bit later. It will increase, suggesting that the baby has noticed the difference. And we can time how quickly he is doing this discrimination and confirm that babies have a really strong talent for discriminating all of the phonemes of the world's languages, and then later on select the particular ones that are used in their native tongue. A little bit later, again with my wife, we thought that we could do MRI in little babies. This was quite a challenge, it's still a challenge 20 years later, but it can be done in completely ethically reasonable fashion. We protect the babies against the noise. We try to prevent their motion. We make them happy by giving them a mirror where they can see the parent. And we present them speech. And what you see here is an activation caused by speech, by the mother tongue, hearing the mother tongue in a 1.5 Tesla scanner in MRI. And if you are familiar, you can recognize Broca's area activation as well as a lot of temporal lobe activation. You're essentially following the same circuitry, the same areas as you would in an adult brain. And this means that already at that age, at 2 months, speech is already channeled to the proper brain systems. Of course, babies at that age don't understand speech. They don't have the vocabulary. They don't have the grammatical structures. But the input is already channeled into the proper circuits that are going to be able to quickly learn about it. Babies learn language faster and with fewer data than current AI systems because there is this preparedness for learning language. And we also learned that the prefrontal cortex and Broca's area could be activated very early on. There was this strange belief that the frontal cortex would come online only at the age of 10 or 12 months. It's not true. It's already very active. It's just slow because the baby is not myelinated. So it's a very slow brain, and the reactivity is very slow, but it's already functioning. So relative to that, what happens when you learn to read? When you learn to read, you reuse this language system, but what you do is you connect to it a new interface. You essentially take your spoken language system and you connect to it an interface from vision. It's a sort of sensory substitution, if you wish. You substitute vision for audition. And we map this in this image here. I'm summarizing a lot of work, but we map this by doing experiments where we compared people who have learned to read, like all of you in this room, with people who are illiterate. I take this also as an apartheid to mention that there is too few research into non-standard participants. We always take, collectively in the field, we tend to scan students from the university. You know, that's the easiest. They are available. But what we really need to do is scan people that are different. And here we're scanning people who are completely illiterate. We had to find them. We didn't want to scan people who were dyslexic or something like that. So we found people who just didn't have the chance to go to school. We found them in Brazil and in Portugal. Relatively old people. In Brazil, they could be younger, but in Portugal, these were essentially old women whose family didn't have the possibility of sending them to school at the time. And so you can see on what we were doing here, we're correlating brain activity with literacy. And you can see that in all of these areas in colors here, there is greater activation to a written sentence as a function of literacy. So what do we see? What we see is, first of all, that with literacy, you will activate the same spoken language system that you would get from spoken input. You activate the language areas of the superior temporal sulcus and temporal lobe here, and also Broca's area. And you will get the same amount of activation from vision as you get from spoken language. But you also activate the visual system, and this is my favorite area, the visual word form area that we discovered with my friend Laurent Cohen, or that we explored. There were a few precursors. This is in the left hemisphere, a region that specializes for the written word. And we also found that it will connect to the spoken language system in a precise manner. There is this area in the superior temporal region here, which is activated now for spoken language. So it is really a speech area, but it responds even more as a function of how much reading you can do. Its activity almost doubles as a function of the reading score, which is the number of words you can read per minute here. So think about it. When you're learning to read, you're changing not just your visual system to tune to the shapes of letters, you're changing your spoken language system to distinguish phonemes better, and you're interconnecting the two. And we also found that the connections between the two have been changing. So I think this was important work to begin to show how much plasticity of the brain is available to learn completely new cultural functions such as reading. Of course, we wanted to do that as well in young children, and I just want to show you this is completely feasible. This is already from a few years ago. We have a mock scanner. We call it the 0 Tesla scanner because it's just made of wood, right? There's nothing inside. It makes the noises of a real MRI and allows us to train the little subjects that we recruit to the lab. They are typically first graders or kindergartners. They are taught that they are astronauts and that they explore other planets. They have the buttons and the coils and everything. And they are told that if they move, the spaceship will not go in the right direction. So they really should be very still. So here's our little statue ready to be scanned. With a little preparation like that, we get very good data. And we were very lucky, it's just one of the studies, but we were very lucky to get permission to scan the same children, 10 astronauts, every 2 months. So here you have 2 scans before school. This is school entry. And now you have the scans as in first grade. We are very serious in France. This is the age when you learn to read. Very quickly, you learn to read. And so you can see the emergence of responses to written words in this child as a function of a few months of schooling. And we discovered that the visual word form area, this visual area that responds to written words, is already activated right from the start after just a few weeks of training, and of course gets larger with training and gets more concentrated with automatization. And this was completely compatible with this idea that this visual word form area connects to the language system, and that the connection between graphemes and phonemes, letters and sounds is the key to learning to decode speech and the key to learning to read. This is what we can get now with a 7 Tesla scanner. This is a single subject, not the average of multiple subjects, as I was showing before. We saw that there was a visual word form area. We discovered that this is not one area, it's little patches of cortex that are ultra specialized for words. So you can see some of this specialization here. This particular voxel will respond to words in English and words in French in a bilingual subject. It will not respond to any other category. It will not even respond to numbers. Numbers can be very similar to letters, right? But because they serve different functions, the brain separates them. And to a large extent, we can find voxels that are uniquely tuned to letter strings. So essentially, I think it's a metaphor a little bit like astrophysics. We thought that there was a single planet here, the visual word form area. We're discovering that it's a galaxy. It's multiple patches of cortex that are more specialized than we thought. And what you can also see on this slide is the range of stimuli that we were using here. So we are presenting all sorts of strings of letters to the subject. This is a real French word. This is a real English word. This could be an English word, perhaps. But as you go to the left, we violate more and more the statistics of the languages that we are testing. And all the way to the left, you know, I think you can agree that this cannot be a possible word. Maybe it could be a word in Polish, but not in English. All right, I don't know Polish. So what you see here is that for the occipital cortex, for the early visual regions, it does not matter. All of these are valid visual stimuli. They're made of little strokes, right? But as you go forward in deeper into the visual cortex, like 3 and 4 here, you have this very, very strong gradient, which means that these areas become tuned to the statistics of your language. It will not, at this stage, know whether it's a real word or not, but it will know whether it's a plausible word or not. And I think that's a good metaphor. The brain is a statistician. The brain will extract the statistics of the input and encode them. And I just mentioned that in passing, but I've become more and more interested in education. And I think, you know, we know enough about reading to make very precise recommendations about how reading should be taught, because brain science is meeting directly with research into the classroom, research into which methods work best for children to acquire reading. And that's why I have now this subgroup in the lab which is developing software and actually a whole reading method that is being used in first grade classrooms in France and now more and more in South America as well, implementing all that we know about the science of reading and accelerating the learning of reading in young children. But in the last few minutes, I want to go back to this slide that I showed at the beginning. The promise was, could we actually decode the thoughts or mental representations of people inside the scanner? Can we do that? In principle, if there is, as I was trying to show you, a systematic mapping between brain activity and cognition, well, in these experiments, we manipulate cognition and we look at brain activity, but in principle, we could reverse this mapping. We could look at the brain and determine what was the corresponding mental representation, right? So this starts to be done. This is an early image that we did with our colleagues, Bertrand Thirion and others at Neurospin in 2006. The idea is very, very simple. In the back of the brain, in visual cortex, you have these retinotopic maps that have a systematic map of the retina. So if you show an expanding ring, the expanding ring is mapped by these different colors onto the cortex. And if you show a rotating wedge, it's also being mapped, first in one hemisphere, then in the other. And inside the hemisphere, you can see it spanning the retinotopic map. Once you know this, it's very easy to make an artificial intelligence algorithm that will just paint an image corresponding to the activation that you see in the brain. For every bit of the brain, you can paint a little bit of splotch of paint on a picture that will correspond to the mapping between the retinotopy and the image. And so what you see here now is not real stimuli, it's a reconstruction of what the subject was seeing when he was seeing the expanding ring, and you can see the expanding ring. And this is a rotating wedge. From brain activity, we can paint a picture of what the subject was seeing. And we could actually do this even with mental images to some degree. If you imagine a T, we could reconstruct a letter T much better than chance. Now, that was the early beginning. Fast forward to 2019, and this is a paper by Kamitani and collaborators, where you can see how much better it gets when you use actual AI to reconstruct these images. And you use not just retinotopy, but all that you know about these visual areas. So, you present an owl and you get sort of fuzzy animal like this. You present, let's say, a glass panel and you get some kind of matrix of colors. It's not bad. It's resembling. That was a very important paper. You can see the letters neuron presented one by one. This was published in the journal Neuron. And so you could decode individual letters from the cortex of the subjects. But now, this is 2023. And with the help of a lot more AI, the AI is capable of generating an image of what the subject has been seeing. So on the left, you have the actual image that the subject was seeing. And on the right, you have, for 4 different people, and for the average of these data, you have the reconstruction by the AI. And I hope you can see how spectacular it is. This image is not in my book because it's too recent, but it's really spectacular. So for instance, the skier here, there's always a skier in the snow in the reconstructed images from every single subject, or the bathroom here. It's sometimes a little bit funny, like this is an animal through the door, and you can see that the animal is a bit distorted and so on. But this is quite extraordinary. We're generating an image of what the person was seeing from brain activity. We can discuss, I'm sure there'll be questions about that. I want to make sure that you don't believe this can be done in an airport when you pass, and people will decode your thoughts or the number of your Visa card or something like that. This requires several hours of someone voluntarily sitting still in an MRI and contributing to the experiment. So I don't think this is going to be done against the will of a person. But with the will of a person could be very, very helpful, for instance, for people who are paralyzed and so on. And of course, the question arises, could you write into the brain? If you can read into the brain, can you write into it as well? And there is no reason why you could not. You just have to find the right techniques that are not too invasive and so on. This is an early paper by Peter Rovsema and collaborators where they did exactly that. They used retinotopy again. So there is a whole array of these electrode arrays here in the back of the brain in the retinotopic areas. Each of these green dots is one electrode. Each electrode was mapped to a particular location on the visual screen. So they are all in the bottom right quadrant here. And then the real experiment begins. You stimulate just the electrode in red here, okay? And they are selected because they form a round shape, right? The shape of a letter O. And the monkey was trained to report which letter he was presented with. And the monkey makes a saccade to the letter O. Okay. If you stimulate a T, the monkey will go to T, and so on, and L. Almost 100%. So you could paint an image on the surface of the cortex by stimulating and getting the right coordinates for these little bits of paint. So this is where we are now. And this last slide is actually my own brain. It's a 3D reconstruction. It was scanned 20 years ago. It's nice to have a backup. It's, for any of us, I think, in this room, it's hard to fathom that my entire personality and all my knowledge can fit into these 3 pounds of flesh. Does my entire self really fit in? And several of my friends with whom I discuss this find this vision deeply upsetting. According to them, the idea that there is no soul, no autonomous self, can only lead to depression, nihilism, or paradoxically, selfishness. I understand those fears. But after pondering these issues for years, I have reached the conclusion that it's perfectly possible to be a lucid, happy, and sociable neuronal man. I think neuroscience is just the avatar of the ancient injunction, know thyself. And seeking to understand everything around us, including ourselves, is a fundamental human quest. We are all in search of meaning. And unless we have no respect for human reason, we cannot turn our backs on the answers that it yields about our own nature. And I think Louis Thomas already discussed this in his best-selling book, The Medusa and the Snail. The feeling that we possess a single self is largely illusory. We now know that our bodies and brains are just a clumsy hodgepodge of disparate cells and circuits, and they don't always pull in the same direction. We are multiple cells. We are a complex system. I would also argue that acknowledging our neuronal nature is liberating. Forget the inaccessible longing for perfection, which crushes us with guilt, especially if you are, like me, from a Catholic family. We are just the lucky inheritors of hundreds of millions of years of the most sophisticated evolutionary learning algorithm, followed by thousands of years of cultural fine-tuning. We are a fantastic machine. We are a machine, but it's a fantastic machine. And accepting our neuronal nature is a lesson in humility, in tolerance, and in humor. Humility because we do not decide on our neuronal balance. With a single stroke or brain alteration, we can sink into depression, any of us, schizophrenia, dementia. Tolerance, because the burdens that affect others tomorrow may become ours. And humor, is the most important, I think, especially in these days. Humor, because considered in the eyes of the naturalist, human behavior is an inexhaustible source of amusement. Think about it when during your child's nest tantrum, perhaps. Or if you have a teenager, maybe even better. In The Medusa and the Snail, Louis Thomas wrote, I quote, "We are perhaps uniquely among the Earth's creatures the worrying animal. We worry away our lives, fearing the future, discontent with the present, unable to take the idea of dying, unable to sit still. Worrying is the most natural and spontaneous of all human functions." We have not mapped it onto the brain, but we should. Worrying is the most natural and spontaneous of all human functions. It is time to acknowledge this, perhaps even to learn to do it better. I hope that my talk tonight may have helped you on this path. It's perhaps time to stop worrying and love the brain. Thank you. Thank you. Oh, and I guess, no. Thank you. All right, okay, I'm a little late now, sorry.
So thank you, Stanislas. That was a spectacular talk, and I don't think it escapes anyone's notice that the combination of scientific brilliance with philosophical beauty I thought was really compelling. So we have time for some questions from the audience, and there are microphones, so think about your questions. If you have one now, raise your hand, and we will try to get a microphone to you, and we'll begin at the back right here.
Which is more effective to learn which is more effective to learn something, to hear it or to read it?
Both. It's better to have multimodal
not a good
input.
answer.
No, seriously. The best way is multimodal input. But the most important is the depth of processing. So experiments have shown that if you just read something superficially, for instance, just for counting the number of syllables, then you will not remember very well the content. But if you go deeply into the content, for instance, maybe because you have to take some kind of decision about the words you're reading, a deep semantic decision, then you'll remember them better. So there are rules like that I discuss in How We Learn that can make your memory deeper. And by the way, this is maybe one time, there is a lot of mythology in brain research. One mythology is that different people have different ways of learning. That's not true. There is no such thing as visual people and auditory people that would learn better with their own modality. We all learn with very much the same learning algorithm.
Is there a question on the aisle here?
You've ruined one of my favorite jokes, which is that I would tell my friends I went to go in for a brain scan and they found nothing. I'm afraid we can't use that any longer. My question for you involves, I guess, artificial intelligence. You referred to it only briefly, but since the brain is such an astonishing statistician, and since it is capable of doing the nuances of whatever its neurons are perceiving, wouldn't AI be very useful in terms of compounding at tremendous rates this accumulation of statistics to help you to go farther into what it is you're examining?
Yes, absolutely. And in fact, there is a lot of research comparing AI with the brain. It's just not there yet. There are many ways to do statistics. Many of us believe that the brain is a sort of Bayesian machine. So it's not just accumulating statistics bottom up, but it's actually starting with very strong hypotheses, priors, models, and it's applying these models to the world and selecting the best fitting model. So the idea of the Bayesian brain is one of the most powerful in current neuroscience research. Unfortunately, it's not really the one that's dominant in AI at the moment, where there's not so much priors. People are already building some kind of empiricist view of learning. Or it's as if they were trying to recapitulate both evolution and learning, right? But in us, learning is very strongly guided by a lot of priors. I showed you in the case of language, that there is this sort of, Chomsky would call it the language acquisition device, a system in young children which is really targeted to learn language. We don't know if it's as sophisticated as Chomsky would have it, which is really like highly structured grammar. It's probably not as rich as that, but there is very, very strong evidence that there's a lot of innate priors that facilitate learning.
Thank you very much for this wonderful lecture. So I have a question. You mentioned that the first few months of a child's school help develop the reading part of the brain. If somehow the first few months they were disrupted in the school, for example, something happens or the teacher wasn't good or the parent wasn't giving them attention, some family issues, does that affect the future development of their brain for intellectual growth? Thank you.
Yeah, I don't think it will affect it dramatically in such a way that no learning could occur later. What's fortunate is that in the human brain, there's a very large window of plasticity, which is at least something like 20 years, right? So we try to concentrate all our learning, all our education in this period. And even then, after 20 years, we speak now of sensitive period because it's no longer closed as we used to think. It's really reduced plasticity. So being distracted in school is not a good idea for sure, but it will not destroy the ability to learn to read. What's really important is that we can accelerate the learning with better methods. We are not creating, I heard that sometimes you are creating dyslexia by having the wrong reading method. No, that's not true. We're just not giving a chance to children that might be on the border between dyslexic and not dyslexic, and may fall into the wrong side just because they're not given the proper teaching method.
Thank you very much.
Back here.
Thank you so much for the talk. So I was wondering, what is your opinion about the difference between humans and non-human primates? Like, they also have Broca's areas and other similar.
Yeah. So we are actively studying this, and it's a very hot issue. I think it's perhaps the question, human singularity. I'm thinking that it's not just language or language, communication language, but it's actually a lot of languages that are different in humans, including the language of mathematics, the language of music. In fact, we seem to have recursive tree structures in all sorts of domains, including theory of mind, thinking of others. We seem to be perhaps the only species which is capable of recursion in all of these domains. We can think, I think that you think that I thought that, and this is almost infinite. So it's very striking that this happens in all of these domains at once. And it's not just Broca's area in the left hemisphere. The mass network, for instance, is very extensive and bilateral and much more dorsal. So I believe that there's been some kind of transformation of multiple circuits that somehow acquired the ability to do these recursive structures. And we're searching actively for the neural basis of that. Of course, there is a continuity in evolution, but it does not mean that there could not be a threshold where the machine suddenly becomes much more universal, a bit like a universal Turing machine, right? You suddenly, you cross a threshold. So that's a little bit what I'm looking for. Yeah.
Question here?
Yes, thank you so much. I'd always been taught that men and women code language on average differently, like, and also that maybe left-handed and right-handed people process reading and language differently. Do you find those differences or do you not?
Very little. So men and women, no, very, very similar. I mean, if you do large-scale statistics, you might find some statistical difference. It's still controversial, actually, for language. There's enormous universality in what I'm showing you today. Not just for spoken language. Ev Fedorenko at MIT has mapped 45 different languages, all sorts, all families of languages. They all map onto the same brain system, always in the left hemisphere in 96% of right-handers. Reading, it's even more surprising, perhaps. Reading Chinese still maps very much onto the same circuits as reading alphabetical languages like I showed today. Yeah. Handedness, a little bit different, but even when you're left-handed, you still have about 2/3 chances of having language in the left hemisphere, like the vast majority of right-handers. So left-handers are not symmetrical of right-handers. It's much more complicated than that.
A question there, and then a question. Let's see.
Hello, can you hear?
Hello.
When we look, oh, sorry.
Question here, then you. Thanks.
So, okay.
It's okay, you can go first.
Somebody take the reins.
When we look at clouds, which seem to be random, we often see patterns.
Yes.
When we develop equations like F equals ma, that's a pattern that summarizes a lot of data. I have an abstract painter friend who says the mind is a pattern-making machine. Is this possible to research, and have you or others tried to do that?
Yes, well, there are two different things in what you are saying. There is pattern recognition in the visual system of all primates. There's, for instance, I showed you some face responses. In the book, I show some responses of face neurons that are fooled by patterns in the snow or patterns that, we are very sensitive to faces. Neurons are fooled just because they are pattern analyzers. But you were also speaking of a second level, which is scientific patterns, expressions. That, I think, may be much more unique to the human brain. And yeah, we are actively researching it. So for instance, I did recent research on geometric patterns. We humans are extremely sensitive to regularities, even in something as simple as a square. We look at a square and we immediately notice there's something special. The sides are equal, the angles are equal, they are perpendicular. We did experiments suggesting that other primates don't care. They don't see whether it's a square versus a quadrilateral. So we are attuned to patterns. We do science. I mean, perhaps the single unique feature of the human brain is the ability to do science, I think, even more than language.
Question.
Yes. It is very, very exciting to see the technological advancement can help us unleash these possible potentials to elucidate the brain function and the map. And you've covered today for the language and linguistics visual perspectives, but in terms of memory, especially for the cognitive declining individuals, I am very excited to use your technology together with electrochemical mapping to see how the memory is stored, especially the short-term or ultra-short-term that does not require the hippocampal route. But let's say for 5 seconds, individual tried to scroll down the phone, and there is a piece of paper that she has a to-do list. She looks at the phone, but she scrolls down on the paper, thinking that it's the phone. Then that's the test that the brain did not store that information for 3 to 5 seconds. So like the gentleman said, we can map that using your technology in a temporal, spatial temporal map in the brain so that we can track down the route which was malfunctioning and which direction which direction we should go to protect and prevent that delay, that cognitive dysfunction.
Well, these are huge questions. I don't want to overemphasize the reach of technology so far. So it's true that in a hippocampus, it's a fine structure. It has a long tail, just like the animal. Even with high technology, it's still hard to map different functions inside the hippocampus. It's feasible. It's feasible in animals, but in humans, it gets really difficult. And I haven't emphasized the limits of this technology. MRI is wonderful for space, but it's not good at time. So when you're asking about the timing of activation, it won't do it, and below 1 second or 2 seconds. We have other techniques like magnetoencephalography, but they are not good at space, they are good at time. There is a whole array of techniques coming in. What's wonderful about this field, I think, is the interaction with physicists. So we should speak also about ultrasound, which is arriving and yielding very high resolution, and also the capacity to write into the brain, not just to record, but non-invasively without electrodes. I think the future is a marvelous future of more technologies, and you're completely right. It should be applied to disease. That's not my personal work. I work mostly try to understand how the brain works in its normal operation mode. I've worked with patients a little bit like I showed brokers at the beginning. This was the beginning of my career, but mostly using the patients to try to understand the normal operation of the brain. But fortunately, there are many other people, including in this room, who do work on neurological diseases. And this is really fundamental work indeed.
Time for 2 more questions, and there's one over there and one there. Thanks.
Hi, thank you for the wonderful talk. I just had a question, like I guess to sort of in general about multilingual people. Do you see differences in people who sequentially learn different languages, one after the other, versus those who were, brought up in a multilingual like household and learned multiple languages at once? And I guess on a related note, I think you were talking about how when you learn to read a language, it reinforces both visual cues and with speaking. And what happens if multilingual people learn to read different scripts, one after the other, I guess?
We are still doing research into this. The general finding has been that for spoken language, if you are an early bilingual, you use very much the same circuitry. If you're a late bilingual, the second language actually uses more resources, and therefore it actually can be more resistant to brain damage. So it's quite frequent to have brain damage that leaves the second language intact, but not the first because of that. So it's quite surprising. It can be both. When it comes to reading, we are studying it. A lot of the reading happens in the same circuitry, even if you're bilingual. But we found that when we studied Chinese English bilinguals, there were few voxels just loved Chinese and didn't do English. So we're still researching this. It's clear that the shapes of the characters are sufficiently different between Chinese and the Roman alphabet that apparently the visual brain is dedicating different places, very slightly different places to this. But we could only see that at 7 Tesla. If you do it statistically at 3 Tesla, you cannot see it. It's really like a very, very small effect of specialization. Most of the finding is one of universality across different languages.
Last question over here.
You mentioned that we all learn the same way, but how do blind people learn?
Yeah. This is a wonderful question. We are studying blind subjects in the lab, and many people are. I think it's a great example. We are studying blind mathematicians. I don't know if you know, but it's possible to be a very high-level mathematician to be blind, including Nicholas Sanderson was the Lucasian chair the chair of Isaac Newton was blind at some point. And he was a top mathematician. So it's possible precisely because of my previous answer, because we have templates in the brain that anticipate on the external world. And the templates for number, space, time, language, they are very much in place from birth, basically. And even if you don't get the visual input, or if you don't get the auditory input, you're going to develop them in different ways, but still with the same sort of internal structure. So even the blind have normal mathematics. They don't need the visual input. They can get tactile input, but mostly the ideas are already in place, and they need just to grow, basically, from teaching.
Well, Stanislas, thank you so much for your brilliant science, sublime writing, and the brilliant discussion we had tonight.
Thank you so much.
Thank you so much. It was really wonderful.